Experimental evidence for DNA as the genetic material, presented by Subhashri, Udaykumar, and Atulya. What is genetic material? Protein, RNA, and DNA were thought as genetic material, but many experiments suggest DNA as the genetic material rather than protein and RNA. Here, let's talk about Griffith experiment. Griffith experiment reported in 1928 by Frederick Griffith. It was one of the first experiments suggesting that bacteria are capable of transferring genetic information through a process known as transformation. He laid the foundation of the identification of DNA as the genetic material with his experiments on transformation in the bacterium Streptococcus pneumonia. Pneumonia was a serious cause of death, so he was studying the possibility of creating vaccine against pneumonia. Streptococcus pneumonia has two types of strains, smooth strain and rough strain. He used these two strains of pneumococcus bacteria which infect mice. Cells of the smooth strain have a protective layer known as the polysaccharide capsule, which protects the cells against the immune system. This makes the smooth strain highly pathogenic, resulting in the death of the host. Rough strain lacks capsule. It doesn't have any polysaccharide capsule, hence the immune system can easily kill it upstream. Here, the host will be alive. When the cells of the smooth strain are injected into the mouse was dead. Whereas when the cells of the rough strain are injected into the mouse was alive. Next, the cells of the smooth strain were killed by heat. When these were injected into the mouse was alive. Bacteria from the type 3S strain were killed by heat and were added to type 2 or strain bacteria. While neither type 3S or type 2 or caused disease in the mice, the combination was able to kill the mice. In this experiment, he was also able to isolate both live type 2 or strain and live type 3S strains of pneumococcus from the blood of these dead mice. Conclusion. Griffith concluded that some transforming principle was transferred from heat-killed S cells to the type 2 OR cells, which transformed type 2 OR cells into type 3 S cells. While the bacteria had been killed, the DNA had survived the heating process and was taken up by the type 2 or strain bacteria. The type 3S strain DNA contains the genes that form the protective polysaccharide capsule. Equipped with this gene, the former type 2 or strain bacteria were now protected from the host immune system and could kill the host. The exact nature of the transforming principle DNA was verified in the experiments done by Avery and Hershey experiment.
Hello everyone, I am going to talk about Hershey and Chase experiment. Introduction about Hershey and Chase experiment. Alfred Hershey and Martha Chase did the experiment in 1952 that helped to confirm that DNA is a genetic material or not. They got a Nobel Prize in 1969 for medicine and physiology. Why they did the experiment? They want to find out the genetic material is protein or DNA because many scientists still assumed at the time that protein carried the information for inheritance because DNA appeared to be an innate molecule. Before they did the experiment, people thought the protein was the genetic material. They used a bacteriophage virus for this experiment. That blue color referred to the DNA, that outside layer referred to the protein. There was a two sets. First one is protein labeled with sulfur 35. Second one is protein labeled with phosphorus 35. Virus infects the host cell. When it is comes into contact with the host cell, a virus can insert its genetic material into its host, literally taking over the host function. An infected cell produces more viral protein and genetic material instead of its usual product. Once infection was over, use replanted the bacterial viruses. Take the mixture in centrifuge tube and centrifuge it. Check the presence of radioactivity. It was seemed that in the second set of the bacterial pellet showed high high radioactivity. This radioactivity was due to the presence of P32 present in the DNA. Based on this, it was concluded that the DNA is a genetic material.
Streptococcus pneumonia has two types of strain, smooth strain and rough strain. Smooth strain have protective capsule as a result of it highly virulent. Rough strain not virulent. Heat killed a smooth strain mixed with a rough strain and injected to mice. The mice died. Thus something was transformed from heat killed A strain to R strain. That converted R strain to S strain. Every conduct a experiment for find transforming principle. Cell lysate of heat killed A strain treated with a different enzyme and with the R strain is incubated and wait for what happened. Rough strain converted to smooth strain in almost all enzymes, but only DNA failed to converted rough strain to smooth strain. Then the mice is lived. DNA was the transforming principle. In this experiment, the liquid from the tubes that received RNase, protease, lipase and the carbohydrate digesting enzymes was still able to transform the R strain into the S strain. However, the liquid that was treated with the DNase completely lost the ability to transform the bacteria. These scientists carried out the same experiment exactly as Griffith but with some changes and reached this conclusion, that was the DNA is the genetic material for the transformation, so that the mice lived when smooth strain, heat killed smooth strain treated with the DNA. Experiment revealed the chemical nature of the transforming substance to be DNA. With the help of experiment, they showed that DNA isolated from a strep strain bacteria could confer the pathogenic properties to R2 strain bacteria. Two conclusions were derived. Active factor is DNA, which can cause transformation. Still, strain contains the active factor.